Effect of Shigella on microbial corrosion. Do you know that bacteria has been playing a role in corrosion? Yes, bacteria as a microbe and corrosion as a metal. It is a corrosion process influenced by bacteria. Interestingly, this bacteria is able to respire iron in metal to generate energy. Ladies and gentlemen, introducing iron-reducing bacteria. The question is how they undergo metal respiration. Imagine when metal is exposed to water, the bacteria get attached on the metal surface and forming biofilm. They end up reducing ferric iron to ferrous iron from iron oxide compound in the environment, subsequently altering the condition of metal surface. Due to this, they are responsible for corrosion occurrence. They account more than 20% of corrosion failure in oil and gas pipeline. A report claimed oil and gas industry experience $1 billion of pipeline damage a year and causes worldwide economic loss of 2.5 trillion US dollar only to corrosion. The worst scenario cases of corrosion ever recorded was affected by bacteria in 2007 at the Prudhoe Bay of Alaska, just because there was an almond-sized hole in oil pipeline attributed to localized corrosion that lead to loss 200 million US dollar of crude oil spill. The economic impact was not solely on the loss of crude oil production, but the funds for repairing corrosion steel structure, material replacement, and pipeline cleaning up are very expensive, far reaching million to billion of dollars, and extremely dangerous when involving the safety of workers and engineers on site. Another subject of ongoing controversy about iron-reducing bacteria is their role either to induce or inhibit corrosion. Some say they induce corrosion by removing the coating layer of metal surface to further corrode, while others claim that their biofilm protect the layer of metal that inhibits corrosion. So to investigate the effect of iron-reducing bacteria on corrosion, we first prepare these three elements for microbial corrosion to take place. First, the metal from carbon steel. Second, microorganism from bacteria, Shigella oneidensis MR-1. And third, a defined minimal growth M1 media amended with different chemicals. These chemicals would be the variety of electron acceptors for Shigella oneidensis MR-1 to undergo metal respiration. The capability of Shigella oneidensis MR-1 to use both iron and nitrate when oxygen is depleted would be the research objective to study its effect on corrosion for 46 days. The presence of microbes is a prerequisite for microbial corrosion to occur. Here in stage 1, we select the model of iron-reducing bacteria from genus Shigella prior to inoculation in the Luria brittannibroth. The Shigella MR-1 were cultured overnight at 30 degrees Celsius with 150 rpm in orbital shaker. The steel coupons were first prepared into a square coupon before further mounted to epoxy resin and allowed to cure within 24 hours. Prior to microbial corrosion experiment, those steel coupons were polished using abrasive sandpaper until mirror surface was obtained. Finally, in stage 3, the experiments were conducted in serum bottle where oxygen has been depleted. Both Shigella MR-1 seed culture and polished steel coupon were incubated in the test solution amended with either iron citrate, iron oxide, or sodium nitrate as electron acceptor. After 46 days, the color changes of the media solution are slightly different. For sample A, the color of iron citrate had white color solution containing black and thin precipitate. Sample B, iron oxide had black dark brown color with thick solid, and sample C, sodium nitrate had a slight whitish color with no precipitate. To determine if Shigella MR-1 is able to undergo metal respiration under oxygen depletion, both ferrazine and nitrite assay demonstrated the color changes from colorless to dark purple color of ferrous iron and nitrate reduction from colorless to dark pink color of nitrite iron in their respective assay. The incubated steel coupon in each media solution are further examined by using SEM to study the morphology of steel surface. After 46 days of incubation, large intergranular cracks are found on the steel sample with no formation of corrosion byproduct. But yet, there are some unidentified contaminants were observed on the surface. While for the other three media solution, all of them were almost fully covered with corrosion byproduct, consisting Shigella oneidensis MR-1 cultures. The difference between these three is only iron oxide and sodium nitrate form corrosion byproduct, looking like crystal structure in radial shape compared to iron citrate. The corrosion product formed were further analyzed by using XRD. Most of the corrosion products were identified as magnetite and vivinite. From the XRD result, formation of magnetite are the result of dissolution of thin iron oxide layer, release of iron from the steel, and resulting to the precipitation of vivinite. The formation of biofilm on the sample were examined by using FTIR. The FTIR results show that the component of biofilm have the ability to binding to the metallic ion released from the microbial ion reduction activity, therefore forming metal phosphate bond with vivinite in the test solution. I believe that the complexity of microbial corrosion associated with Shigella oneidensis MR-1 to be induced corrosion when incubated with iron citrate. Moreover, from the weight loss result, iron citrate made the highest corrosion rate, followed by sodium nitrate and iron oxide. Our results suggest iron-reducing bacteria inhibit corrosion under the growth condition tested. The effect of corrosion in iron citrate is unexpected due to the high concentration of corrosive citrate ion. Meanwhile, for sodium nitrate, the accumulation of corrosive nitrite is a toxic to Shigella, therefore contribute to corrosion as well. The properties of vivinite got something to do with anti-corrosion aided with the formation of biofilm are suggested somewhat more protective to inhibit corrosion with Shigella. We know that corrosion is unavoidable, but it is a promising result to minimize corrosion using biological treatment.